MEOS creates the ethyl glucuronide and ethyl sulfate metabolites. They are detected up to 80 hours. They are highly sensitive, non-volatile, and considered stable. This allows for a much longer detection window than ethanol. So for example, on Friday night, if you would drink 10 drinks by midnight, the ethanol would not be detected after 10 a.m. on Saturday, but ETG and ETS would be detected up to Tuesday. In detection of ethanol consumption, there are several options, each capturing a different piece of the puzzle. Parent drug ethanol can be detected in urine, blood, sweat, and breath. Direct ethanol biomarkers are found after metabolism from the parent ethanol itself and can be found in urine, hair, blood, and oral fluid. Indirect ethanol biomarkers are produced within the body as a response to ethanol use. PEth and the listed indirect markers here are limited to blood as a matrix. So let's take a look at the two most common tests side by side in urine. Traditional testing used to be for parent ethanol, which was limited to same-day, moderate to high doses, and very recent use with a detection window of around 8 to 12 hours. With the introduction of ETG and ETS, we can now capture a much longer window of up to 80 hours since use. We can see small or large doses, acute and chronic use, giving us a better view of an individual's choice. Both parent ethanol and ETG are known to have potential false positives in the rare case of the uncontrolled diabetic patient. Ethyl sulfate, however, is not subject to these effects, but we will look more at that in the next slides. Finally, a positive ethanol indicates definitive use and potentially the state of intoxication if the testing was for blood and breath. But a positive result for ETG only tells us use or exposure, and it does not tell us a state of intoxication or the level of exposure. So let's look at some commonly asked questions. Is this sample with ETG present and no ETS, is this consistent with alcohol use? My client claims she drank NYQUIL last night before bed. Is that consistent with the levels of ETG we are seeing on this report? My client says he uses hand sanitizer all day long at work. Can that produce a positive result? And the answer is maybe. Unfortunately, it is not always black and white, and sometimes there is a definitive maybe. But we can, however, look at some published information and talk about some considerations to help us navigate these scenarios. So let's take the first one. There is a known phenomenon for the potential for post-collection synthesis of ETG in the urine sample. And that is when a sample that did not initially contain ETG from the person ingesting alcohol, but instead was created within the sample after collection. In order for this to occur, there has to be a very specific set of criteria. In this scenario, the uncontrolled diabetic would be dumping excess glucose into the urine, which would then ferment to alcohol in the sample by microbial growth. If this individual also had a urinary tract infection, it could be providing E. coli to the sample. This E. coli then metabolizes the alcohol and creates ethyl glucuronide. As I said, this would be quite a rare set of combined circumstances. Where the synthesis can occur, so can degradation after collection. In this scenario, the person ingested the alcohol and actually had ETG in their sample. If this same scenario of the urinary tract infection occurred, that could provide the E. coli to the sample. The E. coli now will metabolize the ETG, resulting in negative sample. The very important takeaway here is ETS is not subject to these same microbial effects. This means that ETS will not be subject to degradation or post-collection synthesis. So always consider the presence of ETS as the indication that alcohol was consumed. So here's some examples. ETG at 2400 and ETS at 0 is negative for ETS. This could be from post-collection synthesis. So we have to evaluate further before taking action. Is there glucose present in the sample? Is the person a known diabetic? The next scenario, both is positive above the cutoff. This is consistent with alcohol consumption. And the final scenario where ETG is negative and ETS is present is possible for post-collection degradation, but the presence of the ETS above the cutoff is consistent with alcohol consumption. So let's switch gears a little and talk about the next commonly asked questions, incidental exposures. We should start by understanding that there are a multitude of products individuals are exposed to on a daily basis that contain ethanol, including common household items and even food products. We get many questions specifically about some of these. Vanilla extract, as you can see, has the equivalent alcohol content as hard liquor. Typically, ethanol cooks out of the food when it's heated. However, if you were to drink this in any quantity, it would absolutely be like drinking straight from a vodka bottle. Kombucha tea can contain up to 2% as an artifact of the fermentation process and is known over time to continue to ferment to even higher levels. Non-alcoholic beer is actual brewed beer with the alcohol removed, and as a result, a small portion remains in the beverage. A company can call its product non-alcoholic as long as less than 0.5% of alcohol remains. So theoretically, drinking a whole six-pack of non-alcoholic beer could equal one 5% beer. So let's do a little math. A standard drink is considered to contain about 14 grams of ethanol. So you have one beer here at 12 ounces at 5% ethanol is 14 grams. One shot at 1.5 ounces at 40% is similar, 14 grams. And then finally our wine at 5 ounces. Now that we know this, we can have even more fun with math talking about NYQUIL. My client drank NYQUIL last night. Is that consistent with the levels we see on this report? So taking this math, one standard NYQUIL dose at 30 milliliters and 10% ethanol equals 2.36 grams of ethanol. If we look at a 14-gram serving, that means that 6 correct doses of NYQUIL equal one standard drink. Let's look at a study we performed in our lab for NYQUIL. We have 15 volunteers, one recommended dose before bed, and the first morning void was collected. And this is because it would be expected to be the most concentrated sample of the day because metabolites would pool in the urine over the night. You can see here that 13 of the 15 individuals are well under the recommended cutoff of 500 nanograms. And we do have 2 individuals at levels right above the cutoff at 673 and 766. So is it possible to get a positive result from NYQUIL from the night before? Yes, it's possible, but it's not probable. We need to understand that that was their first morning void and would be the most concentrated for ETG for the whole day. In most treatment testing scenarios, this is not the case. Samples are collected throughout the day in the office after hydrating and likely multiple voids prior to the office visit. Also to note, these are recommended doses. If an individual drank more than the recommended dose, these levels could be higher. Finally, these 2 individuals' levels that are so much higher than the other individuals in the study is likely that they were less hydrated than the others and could have falsely exaggerated levels of ETG in their urine. So we need to be very careful around the 500 cutoff and evaluate their hydration status when a test result is near that cutoff. Let's move on from ingestion to incidental exposure. In evaluating consumption versus exposure, we have to be aware of cutoff appropriateness. Current studies suggest greater than 500 indicates consumption and less than 500 could be incidental. We talked about hand sanitizer, NYQUIL, mouthwash, insect repellent, and that's listed here. We also conducted a study on hand sanitizer. Here, 9 individuals participated and applied 17 to 21 total applications over a workday. Urine was collected for a full 24 hours to capture the highest peak levels, and those levels are listed here. You see, once again, All but one individual were well below that 500 nanogram per milliliter cutoff. And that is applying about 2.3 times per hour in an 8-hour shift. Once again, we have one individual that is much higher and could be so far out of the norm because of the hydration status. Here is a compilation of similar studies. I've also added some mouthwash studies for review. Hand sanitizer, 2 squirts every 4 minutes for an hour. Samples got up to 343 nanograms after 30 minutes. Applied every 15 to 30 to 60 minutes, all negative except for one at 62 nanograms. And finally, applied 8 times over 8 hours, all samples were less than 103 nanograms per milliliter. Mouthwash, a 4-ounce exposure for 15 minutes, got up to 300 nanograms per milliliter, and 3 times a day for 5 days straight got up to 120. So you can see that with few exceptions, environmental or incidental exposure will result well below the 500 cutoff. Keeping that in mind, let's look at what known consumption looks like. You will see these levels vary greatly, and that is because every person metabolizes a little different, and they have different eating habits affecting the rate of absorption, different hydration status, and all affecting the levels you see here. You might also notice a couple of these studies of actual consumption are down around the cutoff of 500, but let's go through. 1/2 drink of ethanol in 1 hour at 4 hours got up to 8,400 nanograms per milliliter. Another study, half a drink at 1 hour, everything was less than 1,200, and a half a drink at 1 hour up to 3,700 nanograms per milliliter. At moderate use, 1.7 to 3 drinks of ethanol in 30 minutes. At 1 hour, got up to 5,500 nanograms per milliliter. And at the peak, which is the most concentrated sample, got up to 124,000 nanograms per milliliter. Another one here, 2 drinks, up to 83,000 at its peak. And finally here, 1 to 2 drinks in 15 minutes, 2 times a day for 8 days straight. You see levels ranging from 1,400 to 710,000. So as I mentioned, you'll notice that a couple of these are a little bit lower, down around 600 and 400. So this is where this maybe comes in. Sometimes we won't be able to distinguish use from exposure. Sometimes there will be a gray area that we would want to give the donor the benefit of the doubt, which is why being aware of and the expectation to stay away from the contributing incidental exposures is so very important. So what are our takeaways today? Always use the ETS presence as an indication of use. Never act on ETG only. Contact the laboratory before changing any treatment plan. We have also discovered that NYQUIL, hand sanitizer, and miscellaneous products that contain alcohol can cause a positive ETG result, but used in the correct dose is highly unlikely. So less than 500, possible exposure or end-stage use. Greater than 500, likely consumption. ETS positive at all is a true positive. ETG positive only, suspect of false positive. Dilution matters. When comparing multiple results or evaluating a result around the cutoff, it absolutely matters. A dilute sample will decrease the concentration for all drugs and metabolites in the sample, including ETG and ETS. A poorly hydrated individual will have higher concentrations. I also want to put on here, I think it's important to understand that an ETG screen by immunoassay or point of care cup only detects the ETG and not the ETS. So a screen-only sample could potentially be missing an ETS positive sample, and that would be the situation that we discussed earlier where the ETG degraded. But the use of a glucose test will help you determine if the sample is potentially a false positive due to glucose fermentation. I often get asked about the utility of ETG and ETS in oral fluid. ETG and ETS do not transfer well into the oral fluid cavity, resulting in a window of detection very similar to regular ethanol testing in oral fluid. It also requires an additional collection device, so can be cumbersome to the collection and testing process. That said, at this time, studies suggest that no additional value can be gained by an ETG-ETS oral fluid test over a standard ethanol oral fluid test. Another consideration, if you cannot get your sample to the lab promptly within a couple of days or so, we recommend you refrigerate it, as ETG degradation can be expedited by high temperatures. Just like any urine drug test, this is not a biological diagnosis and is a clinical one only. We cannot tell you how much was consumed, how often, or the disease state. Because ETG is a metabolite in urine, like other drug metabolites, the levels just do not correlate at all with disease state or dose. And most importantly, the client's monitoring program is important. Be sure to set clear expectations and have clients be very aware of what to stay away from. There are non-alcoholic containing alternatives available to NYQUIL, mouthwash, and hand sanitizer. And finally, always use appropriate cutoffs, because zero tolerance cutoffs below 500 can be very dangerous. Treatment plans should be realistic to the client's everyday routine.
So today we're going to do something a little different, just to see if you are all paying attention. There's going to be a pop quiz. Don't worry, you won't get graded on this, but we did want to add in a little bit of an interactive part here so that you could see some real world examples and see if you can determine for yourself if what you've taken away from this. So, our first question.
Based on what we discussed today, is the result consistent with mouthwash use? So we have positive ethyl glucuronide above the cutoff and positive ethyl sulfate above the cutoff.
Okay, so you should all see a little poll pop up on your screen. Go ahead and answer and let us know what you think it is. We'll give you a couple of seconds here to answer before we give you the real answer. Okay, it looks like most of you had answered, so looks like the majority of you said B. Cynthia, what is the real answer?
Okay, so the real answer is no. This is not consistent with mouthwash use because ETG and ETS is above the cutoff at a very high level. We've got 5,750 here for the ethyl glucuronide and ethyl sulfate at 770. This is well above our 500 cutoff, so this is not consistent with mouthwash use alone. Next question. What do we do with this result? So here we have ethyl glucuronide at 843 and negative ethyl sulfate. Do we accept this as proof of ingestion? Do we call the lab for backup? Or do we investigate further before taking action? And finally, both B or C.
Okay, you should be seeing the next poll, so let's see what everybody has to say. Okay, it looks like we have most of the answers in. What does everybody say? Looks like most of you are saying D. Cynthia, what's the real answer?
So this one was a little bit of a trick question because both B and C would be appropriate. We want you to call us if you ever have any questions. This might be from post-collection synthesis, so investigate further before taking action. And finally, what is this result consistent with? Here we have ethanol at 138, that's parent ethanol, mind you, above the cutoff. Also, we have ETS present at 1,645 above the cutoff and no ethyl glucuronide. So is this consistent with recent alcohol use, potential ETG degradation, hand sanitizer use, or both A and B?
Let's see what everybody has to say here. Give another couple of seconds. All right, it looks like the majority of people said D. What's the real answer?
The real answer is D. You see here that we've got the parent ethanol present above the cutoff, which, as we talked about before, parent ethanol is only detected in that first day of use, so up to 12 hours. So if you have parent ethanol positive, that means they've recently ingested alcohol. It also could be potential ETG degradation because ethyl glucuronide is not present, but ETS is above the cutoff.